Real kiwi. So in this presentation, anyway, I'll start talking a little bit about plant and food research that we work for, and then I'll get into the European canker work. So plant and food research it has 14 sites all over the country. The largest site would be in Auckland, another big site here in Christchurch. I am based in Hawke's Bay, and Monica is based here in Nelson. So those are some smaller sites. My site has about 60 people, and Nelson 35. Okay. We have approximately 800 employees, of which 650 are science staff. So quite a sizable organization for a small country. The income of the company, about half of it is private contracts and royalties, and the other half is through government contracts. So plant and food research, we have 5 portfolios: breeding and genomics, bioprotection, sustainable production, food innovation, and seafood technologies. Quite a wide spread of areas. The areas I will be focusing on breeding and genomics and bioprotection because for the European canker work, that's the areas that we work on. So I'll just zoom in first, breeding and genomics. Now the breeding and genomics portfolio is a very large portfolio that encompasses a lot of different crops and a lot of different strategies and aims and all that. What I will focus on, however, is just pest and disease resistance of pig fruit, which is led by Vincent Bas, another Dutch Kiwi from Wageningen, where I'm from. And the objective, of course, of the pest and disease resistance breeding is to reduce chemical inputs in food production and to have residue-free fruit and all that sort of thing. So reducing residues, reducing costs for growers, reducing environmental impacts such as carbon footprint, and also spray drift and that sort of thing. So Vincent leads quite a large variety of resistance breeding programs, varying from apple scab to PR sawfly, and of course the European canker one at the moment is one of the bigger programs that Monica and I are involved in. The other portfolio that both Monica and I are actually members of is the bioprotection one, and our main focus is residue-free pest and disease control. So bioprotection, again, there's a lot of people working in bioprotection, working on all kinds of different crops, varying from carrots to wheat to apples to kiwi fruit. And there's entomology, pathology, and all sorts of people in there, molecular people. So, okay, so focusing on apple production in New Zealand. We are only a small country, but we have a few different areas where we grow apple. Hawke's Bay is by far the largest, with more than 5,000 hectares. Nelson is the next biggest area, with about 2,500 hectares. We only have about 600 growers left. There used to be more than double that number not so long ago. So the growers have increased in size and reduced in numbers. However, we produce high quality fruit, an average of 64 tons per hectare, and up to 110 tons per hectare in the intensive orchards. And that equates to approximately 18 million cows per year. So, European canker is an economically important disease in high rainfall areas such as Europe, but also parts of New Zealand. It's caused by fungus Neolectra platysma, which is a wound pathogen. So that brings me to the first topic that I want to discuss today, and that's the phenotyping of apple trees for canker resistance. People involved in this project are myself, technician Brent Fisher, Monica, and Owen Stevenson in Watchmaker, Nelson. So I wanted to make the point that that resistance to canker in apple trees is a highly quantitative trait. And this means that resistance varies with circumstances and can depend on tree genotype, on wind type, on environment, climate, soil, nutrition, drainage, tree health, and also tree physiology, for example, dormancy, or is it actively growing. So a tree that might be reasonably resistant in one circumstance, the same tree in exactly the same accession can be in another condition can be highly susceptible. And we see that in the literature. So the literature is very conflicting about Canker. Some literature that is written in particular countries, they say a particular variety is highly resistant in that country, and then in another paper that was written from in another country, they say that the same variety is highly susceptible. So we know that in orchards, infection can occur all year round as long as there's rain. But the most important pathway in most countries that we know of is still the autumn leaf scar infection with chlamydia. Other pathways are also important, but this is because of the sheer number of leaf scars that are available at that time of the year, and usually there's rain at that time of the year, and usually there are spores. It is usually in most countries it is important. It will probably account for at least half of the new infections. So that's why in our breeding program, when we're looking for phenotyping, developing a phenotyping method, we focus on the autumn leaf scars. So there's an example here, and these are the chlamydia produced on these sporodocia. And then in spring we get these sort of symptoms. So in our glasshouse in Hawke's Bay, we have several different trials, and this is an example of a trial where we are phenotyping a family, so a cross between a resistant and a susceptible accession, and that is because we are trying to map QTLs for marker-assisted selection. So what we do here is we assess actually incidence and lesion size, and we inoculate leaf scars, and we paint just below the leaf scar with some white paint, every single leaf scar that we inoculate, and then just before we inoculate we pull off all the leaves because it's also readily come off, and then we place a droplet of spores on top of the leaf scar. And then 10 months after inoculation you can see the result. This is a resistant variety. You can see absolutely nothing happened to the leaf scar, no infection whatsoever. What you can see is the paint that is coming off a little bit, but there's no infection whatsoever. And here's a susceptible one. The top here has died. A shoot has tried to outgrow the canker, but it's also being killed. Now it's completely girdled and it will die as well. So if we're looking at the disease incidence of the seedling population, we find this sort of spread where approximately a third don't get any symptoms at all, so that they have inherited clearly the genes that code for the resistance in this case, and some of them are extremely susceptible, and then we have the whole lot of individuals in the middle. Now if we plot incidence, so that is the percentage leaf scars that become infected versus the lesion size, then we can see that the ones that are really susceptible, so almost every single leaf scar becomes infected when you inoculate, they always have very large lesions. But when you come to the ones that become interesting, because only a few leaf scars become infected, then we find that there's huge variation in lesion size. So clearly there's more than just one thing going on here, but what we want when we're breeding for resistance, we want trees that sit over here, that if the leaf scar becomes infected, the infection doesn't actually occur, or the infection does not establish. But if the odd one does establish, we want the lesion to remain small and preferably not to produce any spores. So that's why clearly it's important to look at both incidence and lesion size. Another thing that we're doing is screening all the elites and cultivars that we have in New Zealand, because the other method, the looking for QTLs, takes a long time, and we really are quite desperate to find a resistant cultivar a bit sooner than in 20 years' time. So we're trying to find out which cultivars that are reasonably edible and already have a certain level of resistance. So Monica and I, we both have screened a large number of accessions, both in Hawke's Bay and the glasshouse. The reason I do it in the glasshouse is because our plant and food orchard in Hawke's Bay is canker-free, so we can't go and inoculate the orchard because that wouldn't be very good for our orchard. However, in Nelson there's quite a lot of canker, so Monica can do her trials in the field, and so that gives a really good comparison between the two systems as well, so we can validate that our model situation in the glasshouse is actually accurate and compares well to a field situation. Both Monica and I inoculate leaf scars. Another thing that we're doing is looking at different times of inoculations. So our standard method is inoculating in autumn, because that is the most natural conditions, the most similar to nature. But we really want to do more screenings a year than just one in autumn, and then wait a whole year before we can screen some more trees. So preferably we want 2 or 3 that we can push through in a year. So we are looking at autumn inoculation of the same leaf scars, breaking up the leaves, the green leaves in autumn, and comparing it, and we found that there's not a lot of difference, so we can probably do that as well, but it's something that we're still working on to confirm these results. So the reason I'm showing this slide here is because it's a relatively simple trial with not too many trees in it, and so it's really easy to see what's going on here. So we have here Blenheim orange being most susceptible. So this is average lesion size we have here. Blenheim orange, the largest lesions, Royal Gala, a close follower, and then we have here Jonathan and Bramley and Grenadier, more smaller lesions. Now, what we try to do, because we measured the lesion size over a period of time, so every two weeks I think we measured it. So that means we can analyze the data using the area under the curve statistic, and also just by looking at each individual time point. And we found that if we look at the area under the curve, we actually find no significant differences at all between these different trees. Whereas if we're looking at 17 weeks, just looking at 17 weeks, or just at 14 weeks, or just at 12 weeks, we find that there are significant differences. And so I believe that this is possibly the better way of looking at the data instead of looking at the area under the curve. Especially because these results, when we repeat the trial, they are usually very similar in the different years. If we're looking at percent infection, so at incidence, for exactly the same trial, you can see that the graph looks slightly different. And in this case, because all the lines are so straight, so we're finding that almost immediately after inoculation, the number of leaf scars that become infected are already visible. They have visible lesions, and over time it doesn't change much. So the lesion size increases over time, but we don't get a lot of additional lesions that become visible over time. So for that reason, the area under the curve here did give us significant differences between the accessions, but again, if we looked at individual time points, we had better resolution. The same trial again, just to show you how, so that we have incidence here and mean canker size here, just to show you how the different accessions behave over time. And you can see that Blenheim orange here, the highly susceptible variety, starts off with almost immediately most of the leaf scars that we inoculated expressing and becoming showing canker symptoms, and then just increasing in size over time, but not increasing in number of lesions. But something like Royal Gala needs a bit of time, and you can see that over time more and more leaf scars become show symptoms. So it is important to do this, and you cannot assume that after 2 or 3 weeks, or 4 weeks or 5 weeks, whenever, when you see the first symptoms, that's good enough. You have to for each cultivar, for each accession, check how long it takes before all the lesions that have become, all the leaf scars that became infected, that they have expressed. So again, a caution that canker resistance is a quantitative trait. Something really important to keep in mind, because we're looking only at leaf scars here. And we know that it's not the only important thing for resistance breeding. We know that because we have a highly susceptible cultivar in New Zealand. And every grower knows that this cultivar is highly susceptible. When we plant it and it gets canker, it just, canker just gets out everywhere. But when we test it in the glasshouse and in the field, we find, and we inoculate the leaf scars, we find, well, it's somewhere in the middle. It's not resistant, but it's not terribly susceptible. So what's going on here? Clearly, this highly susceptible variety, this is not the only thing that's going on. There are more things going on. So the caution here is that if leaf scars are reasonably resistant in a particular accession, it doesn't mean that other ones are not susceptible. And it doesn't mean that the leaf scars remain resistant if the circumstances change. So, for example, if you put a lot of nitrogen on the trees, or there's bad drainage, or the tree health is not good, the tree is sick for whatever reason, then those same leaf scars that in an ideal situation appear to be quite resistant might become susceptible after all. And another thing that we are hypothesizing is that the fungus might actually grow within the tree. So in some trees it might be more able to grow within the tree. So if it infects in one spot, it just grows without any symptoms, grows somewhere else in the tree and just pops out. So these are just important things to keep in mind, and when you think you've got a resistant tree, it might not be. Another thing we're working on is developing fast phenotyping methods. The reason is that our breeders want to develop a resistant tree as fast as possible. So what we want to do is, first of all, increase the number of trials per year. So we want to not just do it in autumn, but we also do a few in summer. So that means we have to compare the green shoot inoculation with autumn inoculation, and we're doing that at the moment. We have to identify the optimal assessment time, because if there's no reason to keep the trees in the glasshouse for half a year, then why would you do that? If you can take them out after 12 weeks or something, then it's a good idea to pull them out and change them. So we have to determine exactly when we can do that. And the other thing that we're looking at is inoculation of detached shoots. So cut them off and put them in a vase or something. The Swedish group have done quite a bit of work on that, and they have found that it doesn't correlate very well with the real situation of a tree with roots. So it's an area that we have to work on, because clearly we can push through a lot more cultivars and a lot faster if this method does work. The other thing that we need to think about is for the breeding work is that breeders, when they breed a tree, they don't come up with a nice mature tree, because for the first 3 years or something of the tree's life it's a juvenile tree, and juvenile wood has different characteristics to mature wood. So breeders don't want to wait 3 years before we can go and screen these trees for resistance, because it's a lot of waste of time. So we are comparing juvenile wood with mature wood. We can do that because Richard Bolt Institute has developed a method of growing trees very fast, and his trees, he can grow trees to over 3 meters tall within 1 year, and so those trees have mature wood at the top and they have juvenile wood at the bottom, and so at the one tree we can compare leaf scars at the bottom and leaf scars at the top. So that's what we're doing at the moment, to check whether we can use juvenile wood for our screening methods. Again, we're looking at detached versus rooted trees, because when the breeders plant out their tens of thousands of trees, what we want to do, we want to develop a method where we come in, take the top of the tree off, which is the woody part of the tree, keep the bottom in the ground so it stays canker-free, take the top into the glass house, put it in the vase, inoculate it, see how it handles canker, and if it's good, then they can keep the tree on the ground. If it's no good, pull it out. So, but it is something that we need to work on to make sure that we actually get accurate results. We don't have too many type 1 and type 2 errors. And of course, the other thing we have to work on in this area is that there's no replication when we're looking at just seedlings. So this is an important question. How resistant does a tree actually need to be? It's two reasons why I'm asking the question. The first one is if we want to breed a resistant tree fast, and so that means we use our elite cultivars or trees that already have some nice edible apples, they probably are not going to be completely resistant. They will have a level of tolerance to canker. So we need to know what level is good enough. We know that Gala is not good enough. So what level is good enough? What level can we cope with in an orchard situation if we have just a normal spray program? The other thing is important here is that the gene pyramiding. So and the use of fungicides is something that perhaps we want to encourage. If we have a tree that apparently is completely resistant and growers then think, we don't need to use any fungicides, the pathogen is more likely to overcome that resistance. So it might be another reason to actually not have a tree that is completely resistant, but still needs some fungicidal input. Okay, next presentation, unless there's any questions about the first one.
One quick question. When you have a tree that shows partial resistance, so you have some regions that develop the canker and some that don't, have you done any work sort of comparing those two sites, maybe the expression level, to see whether it's the canker or whether it's the tree that's determining whether it grows or not?
We haven't, but it would be very interesting. Yes, yes. We are very much wondering what it is about, why does not every leaf scale become infected? And even in some of the really susceptible varieties, it's the question everybody is asking. Yeah. So this is another area that we're working on, and this is working on a pathogen rather than a plant. So we've been doing mating studies of Neometra verticilla. When we're looking at the literature, it's contradictory. Some papers say that canker, the pathogen, is homothallic. For example, in Florida, in France, and in Canada, papers have been written about their local canker population, and they all say it's homothallic. So that means that it can produce parathecia or the sexual fruiting bodies by itself. In Germany, Krüger wrote a paper in 1974 saying, no, it's heterothallic. So that means it needs two mating types, like a male and a female, to produce the parathecia. So we came to this and we thought, well, what would it be? No idea. So we looked at the New Zealand population to determine the sexual behavior of just 11 New Zealand isolates to see if it's homothallic or heterothallic, and if we have mating sites in New Zealand. So what we did is we just chose 11 isolates basically randomly, but we did check to make sure that they were very different and that they came from different parts of the country. So you can see the enormous variation in canker that just in the isolates that we have. Some of them, like this one here, they do this sectoring thing, and they produce a yellow secondary metabolite. Whereas others, like this one here, are completely white and they don't do any of this sectoring behavior. We've also tested all these isolates for pathogenicity in the glasshouse, and an isolate like this one is highly pathogenic. There's an isolate like this one is not very pathogenic at all. So there's a huge range of pathogenicity as well. So, just go back. So what we did in this trial is we crossed every single one of these 9 isolates with those 2. And we also crossed every single one of those with themselves. We tried 3 different media, again based on the literature that we read. And what we tried is apple leaf agar on water agar, glucose yeast agar, and applewood malt agar. So we got, in this case here we had Rologala wood, and in this case we had Rologala leaves. We just cut into small pieces. The wood pieces we then split lengthways, and then we just oorsklaved it twice and put it on the agar. And then we placed 2 mycelial plugs on one side and 1 mycelial plug on the other side. And then the theory was that this one would grow in this direction and these 2 would grow like that, and they would meet somewhere in the middle on the wood, and over here on the leaves, and over here just somewhere in the middle on the agar. So we did that, and you can see here clearly these 2 are the same isolate, and that's a different isolate, but we also did it with themselves. So we had 3 plugs of the same culture on the same plate. We were very excited with the results, and you can see here after just 6 weeks we got some beautiful Parathecia on the apple wood, and it took quite a bit longer, but on the apple leaves we also found Parathecia. We looked at the little blobs over here, and yes, they had S-side and ascospores. The ascospores were viable, and we used them to infect trees in the glasshouse, and they caused fires. So the method clearly works. If we look at the different isolates, we found that this isolate only produced Parathecia when crossed with that isolate. This isolate only produced Parathecia when crossed with the other isolate, and so forth. So we found 5 isolates that managed to produce Parathecia when crossed with one of the 2 isolates. None of them produced any Parathecia when selfed, and none of them produced Parathecia when crossed with the other one, if it produced Parathecia with the first one. Looking at the different media types, clearly the one here, the glucose yeast agar, was not very successful. We found no Parathecia at all. Apple leaf agar was reasonably successful, but in one case there was no Parathecia formed, and it took quite a bit longer before they were formed. So apple wood malt agar was clearly the winner, and that was the method that we continued with. So the interim conclusions of the work so far were that glucose yeast agar, no Parathecia, apple leaf agar, they were present, but this was the best method. Also, the isolates are probably heterothallic, and for 7 of the 11 isolates we found a putative mating type. But we just wanted to confirm this, so filling in the gaps. We crossed all the isolates that either mated with one or the other, and crossed them all in all combinations that were possible, so we could predict where Parathecia should form, depending on where they formed, in which combination they formed before. And when we did that, we found that in every case where we predicted that Parathecia formed, they formed. In every case where we predicted that they didn't. We also optimized the apple medium, the apple wood medium, and we tried it on a couple of different agars, and found that the glucose yeast agar with the apple wood pieces was the best agar to use, because it was just less fluffy and more easy to see the Parathecia. The other thing we did was look at the ascospore progeny. So just from one Parathecia we isolated a number of single ascospore isolates, and for example in this case here we have this isolate here that makes the yellow secondary metabolites and does the sectoring, and then we crossed it with this isolate here that doesn't make the yellow secondary metabolites and doesn't sector, and the progeny you can see segregates out very nicely in those that do produce the yellow metabolites, and those that don't, and those that sector, and those that don't. So this is again evidence that a real cross happened, yeah. Then we had a look at some more Parathecia to see some isolates that we already had from different Parathecia where we had collected single ascospore isolates just from Parathecia in the field to see if we found different mating types from the same Parathecia. We found in one case that both isolates were the same mating type, and in another case one isolate was mating type 1 and the other was mating type 2. So again, that's what you would expect in the case of 2 mating types. We continued and did another 36 isolates that we crossed with both mating types and self. And Again, we found in the selfing plates no paresthesia. In the mating plates, we found 20 isolates that are mating type 1, 5 isolates mating type 2, and 12 isolates that did not produce any paresthesia. Then we tested these again with another isolate, just to check it again, because we didn't know why they didn't produce it, and then it turned out that they were mating type 2. So now in total we have tested 50 isolates, and we have never seen any paresthesia forming in any of the selfing plates. Approximately half are mating type 1 and the other half mating type 2, and one isolate is still unknown because we forgot to test it in the second round. Missed it out. So basically we have confirmed the heterophilic nature of Lutissima in New Zealand. Thank you.
I work with growers, so you get the grower version of the work we're going to do, or that I have done since the 2.5 years since I'm working on European canker. There's a lot of people involved, so I don't name them all, but you get an impression at the end of the thank you slide. So this is where I live, in New Zealand, and I sort of can look down to the research side, which is out on the flat. So probably what I want to talk to you about is how I see it. You may or may not agree with me, and it might be different from your perspective anyway. A little bit on the biology, spore availability, an idea about the New Zealand weather pattern, tree susceptibility and wound susceptibility. Then of course I talk about agrichemicals, working with the growers. We do our fungicide trials. We work with the elicitors, some coppers, and we also work on application technology. Then of course pruning and inoculum control. So the problem as I see it, and you might start off with one canker, one tree with one canker in your block, and depending what you're doing and how you're doing it, or if you leave it completely uncontrolled, you have various ways of this canker progressing, whether it goes on the 10, that's a 10-fold increase from year to year. The blue one is a 4-fold increase, and the black one is just a 2-fold increase. Now the blue one is pretty much what Wiltshire published for Bristol in, I think, 50, something like that. So there was on Bramley in the UK. The red one is pretty much what we're seeing in New Zealand and probably in Bristol. And depending which cultivars, just different scenarios. So this is the problem, yeah? It's a huge increase from year to year to year. And why I base New Zealand as on the 10-fold increase, we did a trial on 3 different sites, our research blocks, and that was on Jazz cultivar. And we did work with natural inoculum. So we started off in July with 0 cankers. And where we had 0 cankers, in December we still had 0 cankers, yeah? And when we started in July with 6, there were 6 cankers. In December we had 62 cankers. And oops, I went backwards. But when we had our 21 cankers, in December we had 201 cankers. So that's where we had no control. So we got a 10-fold increase from our inoculum reservoir in July and before, because I did monitor before. So biology, that's Nicolas' work, our postdoc. We're looking at spore availability, and he has 2 different traps. He has glass light, and we have sort of like the water trap. And that is really the results from Nicolas. We see that's just the monthly average. So he empties, and we change the slides after each rain event. So, and this is just a monthly summary from 8 traps from 2 orchards in our Nelson area. And as you can see, we find ascospores if we have, when and if we have paresthesia, we have ascospore release every single month. And we also trap conidia. These are conidia also from the glass lights, but our water, our rain traps, give us a similar data, so that correspond very well with each other. So we have both spore types available depending on the canker type. Now paresthesia, you really only found an 8-month plus older cankers out in the field. So to my growers I say you shouldn't have paresthesia in the field because you canker prune, yeah? So you shouldn't have old cankers out there ever. So, and I think in New Zealand very much in terms of what I'm seeing for most growers, in terms of the way the disease spread, we're talking about a corneal problem rather than an estis spore problem. Now down here you see this is the number of millimeters of rain we had in that particular month, the number of rain events, and the number of rainy days as part of those rain events. So that was really bad, yeah? That is our typical weather pattern during leaf fall. So from 0 to 100% leaf fall, and that would be for a Braeburn block. And that's for my Rewalker, where I'm working at the Rewalker weather station, our weather data, and you see we have nice, plenty rain events during the leaf fall period, and for us leaf fall period in Braeburn would be mid-May to mid-June, sometimes end of July, depending how the weather goes and the winter goes. In that year, in 2012, it was around to mid-June, end of June. Now, Cornelia dispersal, as I'm saying, for most of our growers, because they're trying to do good pruning and canker control, we mostly have a cornelia problem. So we did a little experiments looking at rain splash dispersal of cornelia, and this was also using slides. So we do find cornelia up to 3 meters and probably further away. That's what we can trap. And when we repeat that on different experiments, we're finding similar patterns on rain traps as well. So yes, that's what we expected with the rain splash sort of like fungus and spores, that we're getting those patterns. And Silvio from Brazil, he's actually did that with trap plants out in the field, and he found infection from a central inoculum source away 4 meters from the plant. So that would very much agree with what we expect. Now, tree susceptibility, and first here, this is, well, we use a yellow paint pen, and you can see here the canker, the very, very early onset of a lesion, and that's when they're just the same lesion 2 weeks later. So you can have a really, once you get the symptoms, you can have a really rapid increase within a few weeks of those lesions. Those lesions here, I never checked those, but those lesions here, they're able to produce spores, yeah? But I want to go home and check my young ones as well if I see spotted oak on those. So we know tree susceptibility is increased by wet feet and by soil compaction and heavy soils. We know that from the literature. We also know from our own work it's increased by foliar nitrogen. It's also from the literature fertilizer-induced vigor, and probably both nitrogen and potassium. And we also know it's dependent on cultivar, tree health, age of the wound, potentially tree age. We don't know that yet. But I would say yes, I'd say younger plants are more susceptible, and maybe that goes hand in hand with their ability to produce their plant defense hormones, but also potentially because growers like to push them on a bit faster. Yeah? And the physiological state, we don't know, but we also have the feeling that a tree in dormancy, when it shuts down, it also shuts down its defense, but the canker doesn't sleep. Whereas when the tree is actively growing, we don't think we get quite as many infections. At least that's true for us in New Zealand. And that's maybe also related to respiration rate of the plant. And we don't know yet whether we have a sign rootstock interaction. But my personal belief is, and I have no proof for that our super dwarfing rootstock probably restrict the sign too much, and therefore they really don't have the chance to outgrow. But that hypothesis has to be still proven, and I'm very happy to be wrong. So, why I'd say foliar urea or foliar nitrogen is important, we did a little trial where we looked at, because New Zealand the leaf shedding period is very long, we're looking at artificial leaf shedding to shorten that and therefore decrease the period we require to use fungicides. And so we use EDTA copper for that, but the question arose, does it also affect susceptibility? And in the previous years we have done it, we used it also with urea, whether we get an advantage plus minus urea. So it was really a leaf abscission and tree susceptibility experiment rather looking at the effect of urea. But what we did find, the EDTA itself, no matter in what format, liquid or granule we used, didn't really matter in terms of susceptibility of the tree, but what mattered whether we had the foliar urea applied. Now this was sprayed at 5% leaf fall, as we also would add urea at 5% leaf fall for scab control. And we got that 6-fold increase in tree susceptibility. So we told our growers, maybe if they have a scab problem, to apply urea to the leaves on the ground rather than to the leaves while they're still intact on the tree. It's much easier to target the urea while it's on the tree, but with increased canker susceptibility, and most of them actually stop doing it as well. Now that is from what Reini said, our breeding program, a small summary, a very small sort of like selection of cultivars that I put up. And in the field, for my field resistant trials, in terms of plant susceptibility, I have maximum of 3 lesions per shoot. So the results here are expressed as the number of lesions out of 3. Yeah? And this is our most resistant plant that we had in the field. None of them in the field were 0. And I have a lot more lesions than Reini has in the glasshouse. I also work with slightly bigger plants. Then it's Robusta, and our most susceptible commercial cultivar in New Zealand would be the Pacific Beauty, which we have sort of like most 2.5 out of 3 leaves expressing, and of course one of our most susceptible where we really got every single leaf expressing. So there is very much, even with leaf scars, there is a cultivar effect in that with tree susceptibility. That's really all that says. In terms of wounds, we have leaf scars on a chest tree. We have leaf scars between 3,000 and 4,000 leaf scars per tree on our M9, roughly 10 to 15-year-old plants. It's slightly different on an envy or Braeburn tree, but nonetheless, sort of leaf scars on a plant you will have in the number of thousands. Yeah? Picking wounds. Picking wounds we have on the chest tree, we have around 220 fruit at harvest on the tree. And that's also true for other cultivars between the 100 and 300 trees, depending on tree size and rootstock. So we have picking wounds in the number of hundreds. And of course, pruning cuts, we really only have in the tens. On a chest, we probably only make 20, around 22 pruning cuts. On the envies, we would make less. On a bigger Braeburn, we make up to 50. But we are in the tens, yeah? Now, when we do artificial inoculations, and this is based on Braeburn, I get 10 to 20% leaf scar infections. We get 60% of the picking wound infections, and we get over 90%, up to 100% of our pruning wound infections. So susceptibility is related to the wood type. In terms of leaf scars, mostly we're talking about annual growth, but we do have leaf scars around spurs, between 5 and 8 around a spur, also depending on cultivar. And what we have for protection are leaf or fungicides. Whereas with spurs, we only have spurs, yeah, the picking wounds on the spurs. And we have, if you're lucky, you have harvest fungicides. They can use harvest fungicides in Brazil. We cannot use harvest fungicides in Brazil. In New Zealand, we can only use post-harvest fungicides. And of course, for pruning cuts, we have wound plates. And just don't prune in the rain, yeah? And in England, as I said, or in the UK, don't prune in the fog either, yeah? So, leaf scars are important, but picking wounds, because of their numbers and their susceptibility, for us in New Zealand, they're actually probably a bigger problem than leaf scars, yeah? Now, what we also know about leaf scars, we know because our winter, we have the leaf fall in autumn, May, June, they peak in November, December. So we have from inoculation until we see symptoms, we have a 4 to 7 months lag phase, yeah? In terms of our pruning wounds, not our pruning wounds, our picking scars, we probably only have a 2 to 4 months lag phase in terms of from infections to symptom expression. And of course, these ones, our pruning wounds, it's probably only 1 to 3 months from infection to symptom expression. Now, I've learned there's other wounds in Europe. You see a lot of these cankers. We really don't see them. Or the bark splitting or frost cankers, and we really don't have too many frost canker either that I'm aware. Our work is primarily around those two. But what is important for any particular grower and any particular cultivar and any particular circumstance, understand which wound type is your problem for that particular crop. And with European canker, I'm very much talking about a block-to-block difference, even if it's on the same cultivar. It's very much, if you're talking about a conidial pathogen, it's very much what happens within that block determines the fate of the block, which has implications on the management. In terms of agrochemicals, my first task when I came to Revoca was looking at elicitors, and so we looked at that's Bion or Actigard versus salicylic acid, which is just salicylic acid, no control and a controlling captain, and we sprayed those two post-harvest onto the green leaf, and then we inoculated at around about 10% leaf fall, and the captain we sprayed prior to inoculation. And you could see we get a reduction in terms of with the Bion, in terms of tree susceptibility. That was actually brilliant results. And even the salicylic acid, that 20% reduction was significant to the no control. And then the salicylic acid, the Bion being an analog of salicylic acid, they really affirm each other. Yeah? So we're very pleased with those results. And the Brazilians have done now more trials, and we're planning to do more trials the next season. So that is very good. Then in terms of fungicides, we've done quite a bit of work on copper, and we use, of course, unsprayed, we use copper hydroxide at our labor rates as we have registration on fruit or on apple trees. And so for copper hydroxide, the copper applied will be 0.6 kgs per hectare. The copper oxide would be 1.1 kgs, and the oxychloride would be 3.4 kgs per hectare. So that's probably important to know. And we spray these at 5% leaf fall and then challenge inoculated. Now, very clearly, we get a control from captain, oxychloride, and oxide, but not from the hydroxide. Roland Weber in Germany tells me that in Germany they mostly work with hydroxide, and they have very good results, but the hydroxide concentration they use is 1.2 kgs per hectare, which is very much what we thought, what we see here, the trend, that actually a copper concentration rather than a product effect. And Roland Weber sort of like little chat I had with them confirmed that. So if you use copper, use it at the appropriate hectare rate. And what I probably didn't say, use it at the right water rate. When you spray any fungicide for canker control, you've got to up the water rate. We don't get very good control where we use concentrate spraying. So on our field trials, we are between 1,500 to 2,000 liters per hectare to get the coverage and deposition. And I get to that. What was really interesting in that trial as well, that was monitoring natural infections. Then from the copper oxychloride, I also got a control from that one single application at around about 5% leaf fall, that I still got control on all my natural infection that occurred on those plots throughout the season. So that was also concentration, yeah, probably concentration. Now, agrichemical applications, low copper rates will not work, and equally fungicides will not protect adequately if you have very high disease pressure. So inoculum removal will have to go hand in hand with any agrochemical solution. You have to bring down that inoculum mountain. You have to have a high water rate, high wetness, and we use brilliant blue dye with or without captain, sometimes just with water, to visualize spray deposition. And when you have a canopy in foliage with those orchard sprayers, you get this beautiful eddy formation around the canopy, and you get a beautiful coverage of your leaves or your flowers, yeah? Because you have a full canopy and the sprayers are designed to do so. Now, if you have leaf fall and you lose half of your canopy or more, you don't get the eddies anymore. So what you have is a spray shadow. All the product is on this side, but not on the back anymore. And you can easily visualize that with whether you surround clay or any other dye. I like to work brilliant blue food dye because it shows up so nicely. So coverage and deposition is something we're working at, because your growers waste the money and the time if they use a high concentrate but don't get it onto the leaf scales or onto the picking wounds where the product needs to go. Yeah, it's no good to have it on the leaf if you need it on the picking wound. It's not going to help at all. So spray applications, so we're looking at spray applications, and we're also looking at the use of adjuvants. So we looked at the the polymer, the stickers, the rainfasters, the organosilicons. And we actually found in our simulated spraying that Robin Gaskin has done that work is that the organosilicon type stickers, they improve the deposition of captain onto the leaf scars, while we also have, after a simulated rain event, also still have more captain on the leaf scar after the rain event. That is only because, than captain alone, that's only because the deposition rate was so much higher. So in terms of the wash-off was the same in proportion, but because we got more on, more stayed on onto the leaf scars. So we're looking more, currently we have a field trial, results will be available in January, where we looked at a penetrant versus a sticker versus an organosilicon versus, yeah, that's a spreader. So we had five different adjuvants and 4 different mode of actions in terms of slightly reach back, curative, protective, rain redistribution and rain fastness. So that's what we're doing in the field currently. So, canker pruning. You cut to clean wood, which means at least 30 centimeters away from the lesion, or further if you have to. You check what you pruned if you have clean wood, because often canker doesn't come by itself. On the spore trapping Nicholas does, we also see a lot of watery spheria, so you might as well cut out all the canker and all the internal growth from whatever wood pathogen there is. So always cut into clean wood, then we paint and you remove the inoculum. You don't leave it on the ground. And really, when to prune canker? Any sunny day is a good day to remove canker. You can't go wrong. You see it, you cut it, you take it. And we have good pruning paints in New Zealand, and most growers really use pruning paints for the pruning wound, so pruning wound infections for us is not really an issue. Again, you have to train your staff if canker is a problem. Every single person needs to know what canker looks like, and either for them to then prune it, or for them to identify it and to notify whatever canker supervisors or canker gang you have, for them then to remove it, yeah? But it is very important. We have growers that want to eradicate as much as they can, so they have less than 1% tree incidence. They give financial incentives to every single person that works with them, whether it's seasonal or permanent staff, for them to identify the canker. And they pay up to $5 per canker, because it is cheaper than having a canker gang walking the orchards. But it depends. So we have different strategies on canker pruning and different incentives for different growers depending on the tree incidence where they're at and what their philosophy is. Yeah. At higher canker levels, I personally recommend, and that's what our growers do, they actually have what we call special canker gangs. Dedicated staff that do nothing all year round but to canker prune. And you can also give them financial intensive thoroughness. But it's very important to incentivize the people that work for you to be proud of the work there are and to acknowledge the good work that they're doing. And growers have incentives for thinners, for pickers. So it's just another way to manage staff for around canker management, canker pruning. Now, of course, the more frequently one prunes, there is less inoculum. The less inoculum, the lower your disease pressure. The lower your disease pressure, the better your agrichemical protection. Plus, you can afford to miss some of the leaf scars, because let's face it, with agrichemical sprays, we won't get all 4,000 leaf scars on a tree. We won't get all 300 picking wounds on a tree. Yeah? So it's a numbers game. What we tell to our New Zealand growers in our best manual, we say actually do a canker walk every month, but no less than 4 canker walks. It's before harvest, before leaf fall, a July cleanup, and a December cleanup. And that is based on the data. So this is actually the number of cankers I have cut out on my Braeburn research orchard, or my 2 Braeburn blocks. Yeah, that's the 2 blocks. And then I just use the same data from last year and superimpose it for next year, just to illustrate how many, and these are all been removed, so it's not a cumulative number. This is actually then cankers removed. And we actually get over a 10-fold increase from what we had in July to what we get in December. Yeah, it's over a 10-fold increase, and that is the inoculum between February and March, maybe April if you're into chairs. Well, even Braeburn actually, it's more sort of like between March and May. That would be the inoculum available for.
Winter, because it's the opposite.
Oh, they understand we're on the hemisphere, yeah? You just have to think that yourself, yeah? I can't rethink it for you. So this is our autumn, our picking time, yeah? Thanks for that. So this is how few cankers we have to get those infections on picking wounds, that peak. And this is sort of the inoculum we have to have no peak. And you probably say, why didn't your canker walk between March and July? Because we thought we were going to pull the block, yeah? So we didn't prune anymore because we're pulling it in winter. Then we realized, oh no, we have to do another experiment on it, yeah? So that's why I haven't got April, May and June data on there. But it is that low level results in that high level. And it is, when the growers prune in July, this is after the horses have bolted, yeah? It's after all the infections have taken place. All you're doing is July is cleaning up after the party, yeah? Literally. And the mess that you're cleaning up after the party gives you an indication of how big the party was. Yeah? So if you then just model that out in just some curves, in terms of canker, depending what you do, you're either cycling, even bush pruning or whatever you do, you're either cycling your way up. Maybe you're lucky and you're keeping your status quo, but that's where we want to go, yeah? But it's a cyclical nature in terms of the cankers you see every year. You will always have a peak and it will come down. We always nearly have no cankers in March, but then they come again and they come again, yeah? So, in summary, it matters what you do. It matters just as much what you don't do, how you do it, when you do it, the intensity of management practices, the degree of thoroughness, and staff buy-in. If your staff don't like working with or for you, they don't prune those cankers because it's bloody up and I have to go and get a letter. Yeah, they're not going to do that. But if you give them those intensive and they take pride in their work, they will do that, yeah, or they say, oh, at least mark the tree and tell somebody there's cane crab on the tree. And it's a big problem, and a grower can't do it alone. Not in New Zealand, where the average orchard is between 80 and 100 hectares. I don't know what the average size is here. In Brazil, it's more between 5,000 or 500 and 3,000 hectares. They certainly can't do it by themselves. And there's utterly no room for complacency. Now, in terms of management strategy, that's what we're currently working on, is we develop management strategies at 1 or less % tree incidence, up to 5%, 5 to 15%, and I don't know if those cut-off levels are the right ones to work. Maybe we can group them bigger, but that's sort of like currently our idea, to have a strategy, and the strategy will include a pruning strategy, a spray strategy, a cost-benefit analysis, a life expectancy of the block, as well as an exit strategy. Because if you have 50% tree incidence, or probably a 30% tree incidence, maybe it pays just not to do anything and live with what you have, and think maybe it lasts another 3 years and it's an economic block, and then you pull it out. So these are all ideas and thoughts that have to be played through. Now, as I said to the grower, if you're not happy with the level of canker in the orchard, that clearly shows whatever you have done today has not worked for you, yeah? And so therefore it's time for change. You have to do something differently, or you have to do more of what you have done. So clearly it has not worked. So that's my end. And as you can see up here, this is sort of like a small nutshell, sort of like summary, whereas we have all complete industry involvement, from people from New Zealand to our local consultants, to our retail, to our producers, to our, and all the other people. Thank you.
Thank you very much, Monica. Thank you.